Okay. So now in this chapter, when we are seeing, you can see the chapter here. So they given in the beginning here, they given about some, you can say like the readers. There are some questions here. It tells about. See, let me read on. You have learned in your previous classes that sour and bitter taste of food are due to the acids and bases respectively present in them. If someone in a family suffering from the problems of acidity after the overeating, which the following would you suggest as a remedy? Lemon juice, vinegar, baking soda. This is they are talking about something called a neutralization reaction, which property did you think that while choosing the remedy? So suppose overeating means there is a lack of acid, so we should take the acidic drink like cool drinks or lemon juice you can take. And recall, surely you must be have studied in your knowledge about the ability of acids and bases nullify. This is particularly they are talking about, we can say, uh, so this is about they are saying about neutralization reaction. So we know very well what you mean by neutralization reaction. Okay, neutralization reactions is between acid plus base, acid plus base. So gives rise to salt and water, and along with that heat is going to liberate generally. This is called neutralization reactions. Okay, next recall how this tested sour and bitter taste substance without testing them. So as we discussed here, so they are talking about again here something called indicators, right? We discussed about in seventh class what you mean by indicator. Generally in that class, talk about litmus indicator, and one more we discussed about China rose indicator, China rose indicator. This will let discuss again once again here. You already know that, and the substance are color and litmus blue and will be SSR. Okay, so they are talking about here indicators first, and you are used this is here. Have you noticed that the stain of the curry on the white cloth becomes reddish brown while when soap reddish brown in nature, and this is scrubbed on it turns yellow again when it is cloth is washed with plenty of water. So you can also use indicators such as a methyl orange indicator and phenol indicator. So in these chapters, we'll study about the reaction of acids and bases. So we are going to discuss here about acids and bases. So how these acids and bases are cancelled out. Again, we are talking about neutralization reactions affect the many more interesting things we used and see in our daily life. Okay. Then after this. So this box is tells about some kind of natural indicators. You can see litmus. They are given litmus solution here. They're talking about litmus solutions, which is an purple dye it seems, and extracted with plant is called lichen plant. Belongs to the class division of Thallophyta. It's an division of plant. We can say commonly used as indicators when litmus solution is when the litmus paper is neither acidic or basic, its color is purple. So when it is not neither acidic nor basic, remember original color we can say it's an purple color. So there is an many other natural materials like red cabbage leaves. So it will be shows around 14 colors as per the like something called we have pH and turmeric and colored petals of some flowers such as hydrangea, petunia, and geranium can which indicate the presence of acids and bases in the solutions. Okay, so these all things we can use as indicators. Generally, we'll call as what are indicators. This we are going to discuss about indicators. And after they given about a question here, understanding question, you have provided this. Let me discuss. Okay, so acid and bases. So these are the acids here. Some acids name sulfuric acids and nitric acid, and you should know acetic acid formula. Sodium hydroxide is a base, it is calcium hydroxide is a base, it is potassium hydroxide formula is KOH, magnesium is MgOH taken twice, ammonium hydroxide NH4OH. Okay, so they given here these things, and put a drop of each of above solutions on the watch glass one by one and test with this drop of indicator shown in the figure 2.1, shown in the table 2.1. What change in color did you observe with the red litmus? So they are asking you people to find out the color of red litmus, blue litmus, and phenolphthalein and methyl orange solutions for each of solution taken. Solution taken. Then after they given about this table, and these indicators tell us whether substance is acidic. So we know very well about indicators, what they work and all. Again, they have something called here olfactory indicators. Let us try out of these indicators. Take some plenty of chopped onions. They're talking about we can say olfactory indicators here. Okay. Then after they give about rinse within both and this blah blah they give vanilla, and these are this vanilla, onion, glow, and this is called as olfactory indicators. And after, how do acids and bases react? How do acids and bases react with metal? Let me discuss until here. So let me first let me think about a definition. So this chapter name we can write as acids, bases, and salts. Okay, acids, bases, and salt. Acids, bases, and salt. Okay, first one you should know what you mean by acid actually. So first try to understand acid is a substance which is sour in taste. This is important here. This word sour means Latin name acidus. Okay, acidus. That's why the even is as acid. And generally, acids when it is dissolved in the water. This is very important. It produces H3O plus ions, or we can say H plus ion. This is called hydronium ions. This will produce hydronium ion, or we can say it is hydrogen ion. H plus is called hydrogen ions. We can call this is very important. Okay, and again we can say this. This acids, so you should know this also. Acid will be acid, not acid. You can say acidic solutions. Acid solutions will be there. No? The solutions you know very well as they're producing H plus ions. Okay, H plus ions, which will conduct, which makes acid to conduct electricity. We can say electrical conductance. Will act like something called electro electrolyte. So electrolyte means in a solution form it will make the call we can say it will becomes like a conductor. Let we discuss with the activity later. Then after, okay. So electrolyte means in aqueous form it will conduct electricity. It is called electrolyte. Similarly, you can see here this is about acids. Similarly, we can say about write about bases here. So here, so what do you mean by base actually? Okay, you remember this will be the substance which is bitter in taste. So taste of base will be bitter. And we can say slippery in nature. Slippery, slippery to touch. We can say. So base will be slippery to touch. We can say and bitter in taste. Similarly, this base when we are added to the water, it produces OH minus. Is called hydroxide ions. So hydroxide ions they produce hydroxide ions, or we can say hydroxyl ions also. Hydroxyl, hydroxyl ion. This is about bases, and this also basic solution. Basic solutions also conducts current. Okay, this remember. Okay, solutions means definitely there will be an ions which makes the solutions to conduct electricity. Okay, so now this is about acid and brief about acid and bases. Now let me discuss about indicators. Okay, so first so let me write here what do you mean by indicators. Remember indicators, indicators So remember here, why this concept arises actually. See, some substance are there. Acids means generally we can say taste will be an sour in taste. Okay, so base means we can say base will be base, taste will be what, sir? We can say it will be an bitter in taste. Okay, see, this is when we can say when there is an edible substance is edible, then I can taste them. We can say if it is taste will be sour, we can say it contain acid. Similarly, if taste is bitter, I can say that it contain what's are bases. Now suppose if there is an in the lab, there are some solutions are there. So those solutions are, we can say, non-edible. Non-edible, then is it possible to know taste? How we can come to know taste? It is not possible. So in that cases, to identify those things, we are using something called indicator. So what do you mean by indicator? So remember here, indicators means indicators are, you can say, as you can say, this is also called acid base color indicators. Acid base color indicators. Okay, so among them, first one is litmus indicator. Litmus indicators, again we have red litmus and we have blue litmus. Suppose if we take a litmus indicator, it is available, remember it is available in terms of solution, or we can say in terms of paper also. Okay, so this is all the same thing, it is available. Now we see here, suppose if we have red litmus paper, or we can say red litmus solutions, plus if we are added to the, so suppose here we have a test tube. Here we have test tube. Here we have solution X we have, and for that we are adding few drops of what we can say red litmus. Red litmus solution we added. Red litmus solution. Then this solution's color will be, if color turns into red, or if it can remains red, it is, we can say it may be acid, or it can say, as we are adding red solution, it may be, you can say, neutral. Okay, suppose if this solution's color changes into blue, then I can say it will be definitely base, because red litmus paper will changes into blue color. Sorry, red litmus paper, or we can solution of existence blue if it is basic solution. This you please remember. Okay, so generally we can say we have two papers are there, two paper. Okay, so we can say how we can write, sir. This is, suppose if it is acidic solution, then red litmus paper remains red. Okay, red litmus, this we write blue litmus. Okay, so blue litmus, suppose if it is acidic in nature, red litmus turns to remains red, we can say. If I take a blue litmus, will turns to what, sir? It turns to red. Okay, similarly, if we have basic solution, so why again and again they are saying a solution? This question will be arise, it should be arise. They are saying solution, they are saying solution. Okay, they are not taking only pure acid, pure base. Okay, so if it is basic solution, red litmus will turns into which color, sir? It turns into blue, we can say turns to blue. Similarly, if it is basic blue paper, it remains blue. It remains it remains blue. Please remember here, this is about what, sir? This is about litmus solution, or we can say litmus paper. Okay, litmus solution, or we can say litmus paper. Similarly, if I taken about And one more thing, one more indicators we have one more indicator. It is called now available about turmeric powder. This turmeric powder also you can see in the first part they given about stain of curry fell down upon the white shirt, white cloth turns in reddish brown color because this turmeric solutions will, if it is acidic solution, acidic solutions, it remains yellow only. So you know very well color of turmeric powder is yellow only. Yellow. If it is base, we can see in basic solution, turmeric solutions will be turned into a reddish brown color. So remember here, this is about turmeric indicator. Next we have one more indicator called China rose indicator. This China rose we talk about, we can say hibiscus. You know about hibiscus is also called China rose. If it is acid, it remains as red or we can say pink. Generally, this color is called magenta. So magenta. Okay. Similarly, if it is basic solution, it is basic solutions. In basic solution, it turns into green color. Okay. So these three are there. So generally, these three, which three we can say these three are called. This which ones are we can say litmus, and second one is what sir? Second one is turmeric, and this third one is called what's China rose. China rose indicators. These three are taken from the nature, right? So it is available in the nature. This is extracted from the lichen plant. So you know very well, lichen plant will be there. From that you are going to extract a turmeric powder. Also nature in China rose is a flower. So that's why it is called. These are called natural natural indicators. Okay, so which are taken from the nature. So similarly, we have two more. Many are there. Just as per 10th class is concerned, phenolphthalein, phenolphthalein indicator. One more we have methyl orange. So methyl orange. These two are comes under the synthetic synthetic indicators. Synthetic indicators क्या होते हैं prepared in the laboratory, prepared in the lab. Okay, so let me discuss about their colors. What is the color of phenolphthalein? So this phenolphthalein indicator, so if it is phenolphthalein indicators will show if it is acidic medium, acidic solution. In acidic solution will be an colorless. We can say there is no change in the color. It will be an colorless. We can say if it is basic, if it is basic solution, okay, basic solution, it will be an pink color, then pink color, remember. Okay, similarly we have methyl orange indicator. This methyl orange will show acidic medium means it will be an what we can say pink, we can say red color, pinkish red. It will be an yellowish orange, yellowish orange. Okay, this is about this synthetic indicator. Phenolphthalein indicator is synthetic and methyl is indicator. It's synthetic indicator. Phenolphthalein indicator will show an acidic medium which color, colorless. Basic means pink color, and its methyl will show pink or reddish color in the acidic medium, yellowish orange in the basic medium. This is about color indicators. Okay, try to understand. Again, I'm saying these are called color indicators. Means based on the color. Okay, so these are the color indicators. So what do you mean by color indicators? Are so color indicators. Okay, so based on the work, based on the color. Okay, so we have one more type of indicator called olfactory olfactory indicators. So what do you mean by olfactory indicators? So olfactory indicators means they'll work on the odor, or we can say smell. Okay. So which are those indicators? Are one is onion feel. Okay. Here one more is cloyle. One more is what's that? Vanilla. Okay. So how this will work actually? So how we can use this one? Generally, what they will do. So if this onion peel, they take an activity should take in the cloth this onion peel and after should kept in overnight. And this again this cloth will be taken a piece of this cloth and this cloth will be used as indicator. We'll take test tube and test tube. They'll take solution here. Something think like let me. This is a solution of X. Okay. So इसमें a cloth रखने का. So if this cloth a smell remains if it is acid. Okay. Then if it is X is base तो क्या होगा smell disappears. Clear? So if it is acid, smell remains. If it is base, we can say smell will disappear. This is how this olfactory indicator will be used. Okay. So after this one, they given about okay. This is about olfactory indicators, and after we can say about. So let me discuss about what is next concept actually. So you can see there after this one, this until here they given vanilla extract and onion peel and clove oil. Vanilla is there here. Onion peel and clove oil can be used as a olfactory indicators on the basis of your observations. Do some more activities, understand the chemical properties of bases. So remember here they given here HCl solutions and they take NaOH to understand here. Let me see. If solution is what is the smell of this HCl? So it will be smell remains. Smell remains the same in the case of water HCl. In case of NaOH, we can say it will be disappears. Disappears. Smell will be remains disappear. So in this case, it remains it will be an disappears. Okay. So next, and after they given about this activity needs some teacher assistant. Let me say activity 2.1 to 2.3 and 3. So this activity needs teacher assistant. They given here. We see what is the activity and they're asking the apparatus as shown in the figure. If 5 ml of dilute sulfuric acid, add the few pieces of zinc and granules and what you observe on the surface of the zinc. So pass the gas being an award through the soap solutions. Why are the bubbles formed in the soap solutions? And take the burning candle near the gas and fill the bubbles. What do you observe? Repeat the activity with some more acids like HCl, CO3, and CSSUH. So before going to this one, let me discuss. There is a blue question, I think so. They are saying here, observe carefully here. See, what they given. You have been provided with the three test tubes are there. One of them contain distilled water. Okay, distilled water. Remember, it will be an not acid. It is a neutral that you should know. This is neutral. Okay, and the other two contains acid and basic solutions. So solution they respectively. If you are given only red litmus paper, how will you identify contents of each test tube? So I think you got the question. There is an three. I can say there is an three test tubes are there. So they are given here. There is an three test tubes will be there. Let me try it here. This is test tube one. Okay, and here we have a test tube two. Okay, here we have test tube three. Okay, test tube three are there. Okay, three test tubes are there. Okay, so here we don't know which solution is asked. Which solution is, let me label as A and B, and this is C. This is blue question one of acid. Okay, this is blue question. Okay, now see here three solutions are there, and A, this is B, and here we have one solution will be an acidic solution. One is acidic solution. One may be the one is basic solution, and one will be what, sir? It is an distilled water. So distilled water, remember, distilled water is nothing but what, sir? It should be an neutral. And what they're asking, sir, with us only they available. This water is neutral, available. Only one paper. That which paper they given, sir? They given red paper. They given red litmus. So red litmus or either blue litmus, we know very well. If it is acidic, it will show red color. If it is base, it will show. What about this one? So let me think here. So if they given paper, red paper they given. Okay, let's think like here they given red paper. We kept red paper here. We kept red paper here. Okay, so now red paper they given here. So we kept red paper. Think like this red. This is red paper. This is red paper they given here. Okay, so if it is acid, what will be red remains red only. If it is base, whatever red remains again, not red remains red, it will turns into which color, sir? It is in blue. If it is remains red again. See, try to understand. Again, I am saying if A is acid, it will be red remains red. If A is neutral, also red remains red. Suppose A in the A solution A, this paper is turns into blue. Let you think that. So here we are taking this half of the part which is inside the solutions is turns into which color, sir? Turns into, think like it is turns into blue. So we kept a litmus paper. Which papers are? Red litmus paper. It turns into blue. Then the solution is what, sir? This is nothing but A is basic in nature. It is A is basic in nature. Now see, try to understand here. Very, very important. Now this paper color, what is the paper color, sir? Paper color is what, sir? Is paper color is, remember, half will be red color, half will be blue color. So same paper I'm keeping here. Same paper here. Okay, same paper. Okay, which was half of the paper was what, sir? It is an blue color was there here. Okay, blue color. It remains it blue color, and half of the paper, what is the color, sir? It was a red color. Okay, it is red color. Now see, blue paper again remains blue. Again remains blue. Then what it is nature, sir? It will be an neutral. It is an what, sir? Neutral. Distilled water. We can say it will be an distilled water. Why distilled water, sir? You see here. First paper was red color they given. This is base. That's why it turns into blue color. With me, we have with us with a paper which contain both blue color and red color. So that red remains blue part remains blue. So what we can say, it will be an neutral. Suppose blue part. Suppose if we given same paper again here. Again same paper, which was some part will be an red color and some part will be what, sir? It is again turns into what, sir? Red color. Okay, and what it is, sir? It is an acid. Remember, okay. Again, I'm saying. So now here, three test tubes we taken. Try to understand the question here. Solutions. There is one solution which is acidic in nature. There is another solution which is an basic in nature. One more solution which is a distilled water. Distilled water means, you know very well, it will be a neutral. So now they are taking only one. They are giving only one paper which is red color. Now that paper we are taking in the solution A. If solution A turns red paper into blue, then solution A will be what, sir? It is an basic in nature. Okay, clear? Now, paper color is two colors. One is red color, another color is what, sir? Blue color. This blue part remains blue in the solutions. Then what? It may be base. it may be distilled water. Anyway, already we found the basic solutions kept as A. Now what will be the solution B? Definitely it will be an what, sir? It is a neutral. So suppose same paper which is have two colors, we're keeping in a third test tube C, definitely it should be turns blue color into again what, sir? It turns to blue paper into red color. That's why this is an what, sir? Acidic. Here what happens, sir? Here blue remains what, sir? Remains blue because this is an neutral in nature. Yes, here it is red was turns into what, sir? Turns into blue. This is how we are going to identify the acids and bases and neutral when they are providing only one litmus paper. So this is about indicators. Just remember, indicators are two types are there. Indicators are two types are there. One is not two types, we can say three types also. So indicators are two types based on the resources from their sources. One is called natural indicator, another is called what, sir? Another we can call as synthetic indicators. So natural indicators, which are natural indicators, are we can say litmus paper, litmus solution, turmeric powder, china roses. They are as they are obtained from the nature. We can call them as natural. You can classify them as a natural indicator. Similarly, phenolphthalein indicator and methyl orange indicator. These indicators are prepared in the laboratory. That's why these are called synthetic indicators. Synthetic means a prepared, man-made indicator. We can say artificial, man-made. We can say okay, no? Means nothing. They are not man-made exactly. They will take some chemicals and they will prepare. That is called synthetic. Similarly, so color of litmus indicator will be remember a red color in acidic medium, blue color in basic medium. So you can remember ARBB. Okay, ARBB. A for red, acid for red, base for blue. Turmeric indicator will show which colors are. Turmeric indicator will show yellow color. We can say no change in the color during the acidic solutions. If it is in the basic solution, yellow color will turns into reddish brown color. That's why the stain of the curry. See, you know very well, curry is used to prepared by using the turmeric powder also, as it remains there. So when we are applying base of soap solution, they are saying that soap solutions are basic in nature. So base is there, turmeric powder is there. So basic means turmeric powder will turns into reddish brown. That's why while washing, when you will apply the soap on the curry, as curry contain turmeric indicator, it turns into reddish brown. The while washing, everything is gone. That's why we can't able to see the color. Similarly, Chinarose indicator is also comes under the natural indicator. It shows the magenta, or we can say pinkish red or reddish pink, we can call. It is in the acidic medium. If it is basic medium, it will show the what we can say green color. So phenol indicator will show colorless in the acidic medium, pink color in basic medium. Methyl orange will show what we can say reddish, pinkish red in the acidic medium, and red yellowish orange in the basic medium. Okay. So along with that, one more type of indicators are there, which are called olfactory indicators. These olfactory indicators are not working based on the what we can say color. This is based on the smell. So here they are given the activity, and there let me go for the activity once, not so let me discuss here. This they mentioned here. So you can see here they given the activity here. Take some chopped, finely chopped onion, in the plastic bags, and along with some strips of clean cloth. Tie up the bag tightly and leaves overnight. And you can see in the fridge, should keep in the fridge. So cloth strips can now used to test the acids and bases. So take two of these cloth strips, check their odor. So should first, before the doing activity, should know what is the smell. And after, keep them on the clean surface and put a few drops of dilute HCl. So here there will be cloth will be which is used in the which is kept onion in this cloth. They kept what's the onion peel, and upon this one they are adding the few drops of few drops few drops of HCl. Then what happens? The color will, sorry, smell remains same only, and we can say on the you can see keep them on the clean surface and put a few drops of HCl, dilute HCl, means solution there on the strip, and a few drops of dilute NaOH solutions on the other. So two papers, not two paper, two cloth they are taking, Upon they're keeping what, sir? They're keeping the HCl, HCl. Upon second, they're adding what, sir? NaOH. Then they're asking people to find out the smell. Okay, so if it is acid, smell remains same. If it is base, smell will disappear. So in this way, they used to identify acid and bases based on the odor. So thank you for watching. Let me discuss next part in the next video.